The next speaker is Werner Eberhardt from SAP. Werner is going to talk about real-time processing of genomic and clinical data, and it's himself.
Thank you very much for the introduction. Thank you very much for the opportunity to talk to you today. Let me see whether I can get the presentation up here. Here we are. So what I want to do today is really just give you, from our perspective, an overview about the opportunity and also challenges that we are seeing along the lines of the topic of this session overall. I want to talk about enabling technologies. There's a lot out there you actually might not be aware that really can help you with your research work. And I also want to talk about a few applications that we are developing, that we are supporting in this context. So let's talk about the opportunity first and the challenge. I mean, here you can see, and this is brought from ASCO Cancer Link, you can see, the statistic about the number of participants in clinical trials. And really, it is just a small fraction here, the 3% of the overall patients participate in clinical trials. And that is a challenge to begin with. So basically, we are not learning from 97% of the cases that are out there. And even more so, if you can just if you just look at the number here of the population that is recruited for clinical trials. So obviously they have to follow, they have to meet certain criteria to really be able to participate in a clinical trial. And as a result of that, you can see here, for example, in this little example, clear discrimination with respect to age. So typically people in a clinical trial in oncology are much younger than those that you typically would see as you look into the population on the right-hand side. So that is the first challenge. And the second challenge, it's fascinating to see that, Garrett and I actually used the same slide. And I can promise we did not discuss and agree up front. And I'm very pleased. And unfortunately, we don't have your latest finding here already included, which shows that this is really brand new. But the message is clear. I mean, while in the past we looked at, lung cancer as one diagnosis, with all the diagnostic capabilities that we have today, the picture is going to be much, much more complex. And fortunately so, because again, if we want to follow personalized and precision treatment strategies, and the next speaker, Stephan Brock, will also already talk to this, then you really need to have capabilities that go far beyond to what we classically have been using. And this is really the essence of what I want to talk to you today about. Now, one last point of view here, and again, I'm also showing this as an opportunity here. Many of you here are doing leading research in this area. And this is a basic conclusion from one of the WIN symposiums that were held recently. It's clear that if we want to come to those new findings in light of all the data, the big data that has been that is out there, and Garrett just visualized some of that. You need to have different strategies. You need to have different tools. You need to embrace the complexity. So what we typically do scientifically with our models, we reduce complexity to what we really can deal with and understand. So the idea is, as long as you have the right tools in place, then capture the data, then use the right technologies, then to really make decisions. And there is a reason why we didn't do this in the past, and that is exactly the next slide here. So on the left-hand side, you can see what we are typically doing in research. We are doing a deep analysis. We are taking big data approaches. Many data types are taken into consideration. We go deep. It's a 3-year, 2-year, whatever study that we are performing. But the challenge is with that, it's not simple. It cannot be used across the board. It cannot be quickly done. Maybe you have to basically ask for the data, and a couple of weeks later, you will get the data to do this type of analysis on the left side. So wouldn't it be great if this analysis would be very simple, if you don't have to involve IT to build a subset of your database so that you can really run the analysis? You rather would be able, as a scientist, to do the analysis yourself with no data preparation, no aggregates, no tuning. You don't have to wait for the query result a long time. So ideally, would have the combination of both worlds. And that is really what we want to enable with our technology. So it is. backup try to start here. So this is what we tried. This is what we are doing with our technology. I'm very, very proud to say that some of this technology has been developed also in collaboration with Heidelberg here, with the National Centre for the Tumor Research, the University Hospital here, and the DKFZ. The idea is that we take the data from various sources, whether it's health data from sensors, mobile devices, the electronic medical records, or whether it's genome data, which is basically what I also want to focus on and show you during the talk today, and bring it all together on one platform that is enabled in real time to capture the data and also to analyze data. Then this becomes a completely different environment also for you as a researcher to find new findings based on all the patient data, based on all the complexity that we have seen before. So very often we just don't find the right conclusions, we just don't find the right answers because we shy away from the effort to really drill down. And Stephan will show you later also, Stephan Brock, the next speaker, will show you in the next talk also how the technology can be used in this context to make that happen. So a couple more points that I want to highlight in this context. So if you want to do this, you need to have a rich platform, which includes a number of capabilities. Just want to highlight here text mining, for example. As we know, a lot of the data is in unstructured formats. So typically it cannot be used as such if you want to do your analysis. So what we did here in a pilot project 2 years ago at the NCT, we actually took physician release letters. We took them through our text mining pipeline and just pulled out the biomarker information from those letters. And obviously it's not just sufficient to pull out the biomarker information, you also need to semantically understand, in which context has this biomarker been mentioned. So this is what this technology does as well. And then we can integrate it as a source then for insights for your analysis. Data services are used to then basically take the data from the medical record systems. Predictive analysis library, I just want to mention this in a moment. On the right-hand side, you can see what we are doing. A lot of this is based on our HANA platform, the in-memory platform, as is already. Thousands of customers across different industries are using this. And what we are doing in addition here with this golden part is we are adding specific data models, stored procedures. We are making sure the platform is ready for genomic data as well, and also have healthcare integration service. And what then this allows is that on top of this, our own applications such as medical insights, health engagement applications, partner apps, again, Stephan Brock will talk about that, and a user portal can be sitting on top of that. That's what we want to enable. That is what we mean with providing these type of capabilities. I promised to just say a few words about predictive. Again, I firmly believe, and the predictive library is already built into the system, it really allows you to let the machine, let the computer help you really understanding the drivers for the different outcomes. And this is across the board, but also if you have looked at the diversity of diagnostic and biomarker data that we have seen already in the previous talk, it's a key capability. We are just not able, like a machine, to see all those relationships as quickly as a machine could do this. So in this regard, again, probably another key capability. My apologies for this slide. It looks pretty complex, but it's what we want to highlight here is there is a core of capabilities here in the inside in this platform, but it's absolutely key that it's open. It's open to integrate data from different sources. It's open to integrate content, whether it's your content, whether it's the content that you find out there in public scientific databases. The UI can be extended, so applications can be built on top of that, and the pipeline can be extended. We are working here with DKFZ and Amble, also on some of the next generation large projects, and the ability to connect then external pipelines so that they can play and the data can come together on this in-memory platform so that you can really very, very quickly in real time get those insights is absolutely key from that data that you're processing. And what that means in reality, I mean, obviously, when we're talking about big data, as you know, here we're talking about 15 petabytes that are already have been collected. So by no means would you put 15 petabytes of data on in memory platform. I mean, memory is pretty cheap, but not that cheap. So from that perspective, the idea is that we have connectors, we have technology such as VORA, then to basically make sure that we are intelligently able to connect to whatever technology you're using out there already, like Spark, or if this is a Hadoop cluster with Yarn and HDFS connectors, all of that needs to be enabled. And this is what we mean here with this data integration layer. And the pipeline orchestration here as well, which then can rely on different infrastructures that you're using to really manage your data at scale. Now, I also want to go up in this little chart here and talk about the application integration. So what this will allow you to really analyze big data and real-world data. So unlike what we have seen in the initial chart, we are not just talking about the 3% clinical trials here. We are talking about the 97% of the cases that are in general treated. I mean, the goal is, our objective is to make sure that every single patient that comes here, their record is going to be collected here and will become a source for insights and really better treatments. So attack reporting needs to be possible, and obviously this platform needs to be secure and able to understand, predict, and decide. So maybe enough talking here. Maybe I show you real quick, and I keep this very short because fortunately, again, Stephan Brock will show you another slice of this technology. I will use medical research insights now, the application that you can see here on this chart. Pull it up real quick. So what you can see here is a data set that we modeled along the lines of what we analyzed at the time here at the National Center for Tumor Research. So you can see the different ICD codes for cancer. So if I drill down, I have 100,000 patients here in these cases in this database, and look at a specific one now, C54, corpus uteri. And then again, this is now connecting in real time to database, pulling up the cohort, and again, it's a very simple one in this case that I have in this database, and I know instantly I have about 1,414 cases in here. Now, the beauty is you can now analyze, you can slice and dice this in different ways. You can look at, different parameters, surgery, was a radiotherapy done. Again, we'll see more of this in the next talk. Let me just look at the histology. As part of the primary diagnosis, the pathologist will capture the histology data, and let me just pull out two different forms here, endometrioid and serous, two cases, two different instances that have been basically captured, and now a filter basically on these two selected values. And now what I want to do is I want to talk about what is the core piece of the presentation today, the connection of genomic data. So underneath, for those 1,400 patients, what the technology does, in real time, it reaches out and shows you here on screen the different chromosomes and the variant information along those lines. And we can see here for if you look at the gene variants here in chromosome 17 specifically, we have here a deviation here for the source sample. And we click here onto this chromosome, and then we can see in more detail how this looks like. You can see here this little diagram of chromosome 17. We can zoom in further to really understand, where did the modification take place. You can see here the genes annotated. You can see here different splicing variants, the translations, and I can let me zoom in further, because what I really want to understand is what kind of gene has been impacted. And you can see here TP53 is the one, so not a big surprise in this case. And what you can then do here is drill down further. If you really want to understand, you can I can go on and zoom in, and even you can see here single bases. You can see here the individual amino acids for those different splicing variants. So pretty powerful technology. And let me just end this very, very short demo by then also looking at the patient data. So obviously it is of utmost importance that once you found something, maybe you found in a specific cohort patients of key interest, as long as you are the treating patients, and you have access, the right access-wise, then what you can do is you can pick from this list of patients, you can pick an individual one, and then what you see here is a patient timeline, which is, by the way, configurable. So you can see in those different swim lanes the diagnostic results, the radiotherapies which have been done, lab results will be shown. Takes a moment to pull up here. Hope my internet connection has not been lost. Maybe it did. Okay. So this has given you an idea, basically how you can switch with such a technology from the high-level analysis all the way down to the individual analysis of genomes, understanding what are the variants. And we have seen in the previous discussion and during the introduction how important it is if you want to take big data approach to do this. And I'm very, very proud that, again, this technology has been developed here with the National Center for Tumor Research, and there was just recently received an award. So I want to show a little video regarding this topic, so hopefully it will come across well.
Design competition, the Red Dot Award, the one they took home recently for SAP Theory, well, they scored a second numero dos dots. This win, presented in Berlin, is for excellence in interface design. SAP Medical Research Insight solution allows doctors and cancer researchers to quickly, easily analyze patient data in real time. It was one of the winners among 7,400 entries. It's not just good-looking software, it's beautifully functioning too. Docs deal with complex info and improve patients' lives. SAP's cancer work continues in Kenya. Cervical cancer is the most common cause of death from cancer there. It's easily preventable, but most eligible women have never been screened. SAP developed a new mobile app with HANA Cloud. It digitizes screening. Data is entered locally, accessed, and studied. It reduces the risk of losing or duplicating data that plagues paper-based screening. Heidelberg University Hospital is co-leading the Emerging Technologies in Cervical Cancer Screening Project. It's a one-year study of 800 women done by Moy Teaching and Referral Hospital in Kenya. The app works even with poor internet connections, and offline mode can be synchronized to the cloud with a USB stick.
So there was a second news piece in there, but Megan Meany is really unstoppable, so I didn't try to even interrupt her. So this is again another great application, great example work that we have done here with the University Hospital. So what you have seen before really also got the ASCO excited. So we are building for them, and we are actually releasing within the next month. We are releasing Cancer Link, making it, putting it in productive use. So we are building for doctors, patients, and researchers based on this in-memory technology, similar capabilities that you just have seen. And again, for us, it's a great data point. It's great to see that these enabling technologies are actually used in the real world and hopefully can help to get to new findings and improve cancer care overall. The next step, and again, I just want to touch upon this before I conclude here my presentation. So obviously, ideally, would also be able to use at some point in time proteomics markers to really understand, cancer, diagnose cancer. Very early work. A lot of work needs to be done in this context. By no means, but again, here's just an example of what the work that we did with the Technical University of Munich. So what we're doing here is we are putting together on a 2 terabyte HANA server, actually not too big, 160 cores running in parallel. And the website is public, so you can just go there. 92% proteome coverage in the system has been accomplished. You can use this website. Please go there if you're running the mass spec experiments, really trying to understand your peptide pattern. So in this regard, another great capability, which in this case ended up in in a Nature paper overall. And with that, I want to come to the last slide here. I just also want to mention that we are very excited about these capabilities to use precision medicine and also want to let our employees benefit. So together with our partner Molecular Health, we are running the so-called corporate oncology program for employees, which is also supported by the National Center for Tumor Research here and University Hospitals. So great collaboration here, which again definitely helps our employees to become healthy and stay healthy and at least turn their cancer disease in a chronic disease. So with that, I want to conclude, and hopefully I've been able to give you some insights both about the challenges, how we address them, what kind of technologies we provide, and how we apply it. Thank you very much.
Thank you very much. Very exciting data and machinery. Questions?
Thank you for the talk. And then I have two questions. One is regarding the missing data. That is always a challenge for the big databases. How do you deal with the missing data? And the second part is about the security of the data. If everything is in one institute, everything is okay, but if data comes from different sources, especially in the German data protection era, how did you deal with that if you want, for example, if you want to connect or link your data with the cancer registry data or any other institute?
Yeah, thank you very much for those questions. What I showed you is underneath there is this HANA platform, which is again used by thousands of customers. And as a matter of fact, if you are in banking, if you are in other business, the data, the ability to access data with certain restrictions is at least as prominent in some cases like in healthcare. So what we are simply doing is we are using the mechanisms that the technology itself provides to then give a role-based access. Having said that, there's still work for us to do in terms of making sure that we make this even more granular. But again, all the security requirements, I mean, that was obviously top of mind for ASCO as well. Can you imagine if we bring in data actually from third-party hospitals there, and then there are leaks, people access data which they're not supposed to access? It's a big issue. So obviously we went through all the discussions and reviews to really make sure that we are on the safe side there. And I'm sorry, you need to repeat the first part of your question. I'm not sure I completely got it.
Yeah, the missing data, how does it handle the database and the analyze?
And when you say missing data, what kind of data do you have in mind?
You have several different patients, and each one may miss some part of the data. One may miss the genomic, one may miss the therapeutic and other things. How do you deal with the missing data? Do you impute them, or you just ignore and do whatever you have?
Yeah, great question. So what the technology does, it makes it visible. So unfortunately, I didn't get to this at the end. So in the timeline, for example, if you go to the lab results, I'm happy to show this to you in a break afterwards. In the timeline, you go to the lab results, and if there's genome data available, you can see a little icon with the genome data, and then you click onto it. So it's really kind of tuned for your discovery. So as you rightfully said, so not every time you have the data, you need to also then obviously make sure that you display the quality criteria, where's the data coming from. So what technology does, it at least brings it all together, makes it accessible if available.
There's a question over there.
Thank you also for the interesting talk. I saw your output, and it looked very pretty and very uniform. But the data is coming from all over the world, from different institutions, different standards, and it probably has the problem I have seen everywhere with user-generated data. It is dirty data. So some people would input the same thing in one way, others will input it in another way. How do you deal with these different standards from data sources?
Great question. I mean, so the technology, what it does, it basically allows you, number one, to standardize what comes in, to use mechanisms, standardized mechanisms, those were the data services that you have seen, to then basically transform the data to make sure you reach a certain level. However, you might still, since if the input is wrong or fuzzy and all of that, you will not be able to cope with that. So, but interesting, what happens is once you have the visibility for that, people start thinking. Basically, there's something, one of the first reactions that we received when we showed the database in a smaller circle, when we did this at the NCT, and again, this is high quality data just from here, there was a reaction, the data is wrong. And then we said, okay, let's take a look. And then we literally went with, and you can do this with technology, to the original data. And the data was right, but to your point, the data was not perfectly represented there. So what it does, it gives visibility, transparency. You will see that the data needs improvement, and that generates this dynamic that over time people pay more attention to once they have the output, also to the input. So we believe that besides the ability to also enable your research, this type of work will also improve the data quality significantly. And so it's definitely a good thing there as well. And what we also wanted, since we are working with ASCO, we are working with ASCO also to help drive standards.
But are the people who enter the data and the ones who read it the same ones? Could it be that the people who see this need for transparency are maybe the physicians, and the ones who enter them are the documentation people?
Yeah, another great question. We firmly believe that we need to add value to each one in the process. We need to, even for those doing the documentation, we need to make it easier for them also to see, whatever deviations, to make it easier for them to correct mistakes. If we do not a good job there, then I think we'll fail, because again, you're not putting a lot of effort in providing quality data if somebody else down the food chain really just benefits from this and you don't even know who that might be. So it's a great point. It's key to the adoption and making sure that the data is going to improve over time. Thank you.
Thank you. I think if there are no further very burning questions, in the interest of time, we have to go on. Thank you very much again, Werner.
Thank you.